Hello students, welcome back to our YouTube channel Food Tech Online. Today we are focusing on important MCQ on the protein which were useful for the CFTRI MSc Food Technology entrance exam. So let us begin. Before we move to the question, let me tell you about our ebook which is launched for the CFTRI MSc Food Tech student. It has the major courses like the physics, chemistry, mathematics, biology, microbiology, biochemistry and nutrition, agriculture and the dairy technology and the engineering. And the link of this ebook is given in the description box. So moving toward our first question. What is the first question? That is, this is the finest way to characterize a polysomes. Options are active region for the lipid synthesis, B. Active region for protein synthesis, C. Active location for DNA synthesis, D. Each and every one of them. So, correct option over here is nothing but option number B. Active region for the protein synthesis. Okay. So, basically, characterize of the polysomes means what? Basically polysomes means what? Polysomes is nothing but the polyribosomes, right? That is a cluster of the ribosomes bound to a single mRNA molecule, and this ribosome works simultaneously to translate the mRNA into the polypeptide chain. Okay, means it eventually folds the function of the protein, and this is going to be the active region for the protein synthesis, right? So correct option over here is option number B, right? Question number 2 is, in protein synthesis, translocation begins with the migration of the precursor protein. Options are tRNA from P site to A site, bipeptide tRNA from A site to P site, tRNA from A site to P site, tRNA from P site to E site. So, correct option over here is nothing but option number B, bipeptide tRNA from A site to P site. So, in the translocation, especially involve the movement of tRNA with the grooving polypeptide chain from A site to P site. Okay. So, with correspondence to that, option number B is correct answer over here. Moving toward question number 3, the most accurate description of the process of protein synthesis from the genetic code is option A transcription, option B translation, option C replication, and option D is nothing but the reproduction. So, okay, option is option number B, translation. Basically, translation it is a process by which mRNA which carries the genetic code from the DNA. Okay, so correct option over here is B, translation. So moving toward a question number 4. So our question 4 is, this statement concerning the nature of the genetic code is inaccurate. Question here is inaccurate, not an accurate question. Okay, inaccurate. Our options are universal, B is overlapping, C is commaless, D is triplet. So our correct answer over here is option number B, overlapping. So basically what it is, in the genetic code, what is the genetic code is not overlapping, right? Each nucleotide is a part of only one codon, and the codon are read sequentially, okay, without any overlapping. This is essential for the accurate translation of the genetic information to the protein. So our correct answer over here is option number B, overlapping, right? Moving toward question number 5, translocase is the name for this elongating factor. Option A, EFG, EF2, both A and B, and EFTU and EFTS. So our correct option over here is nothing but option number C, both A and B. How it is? C, both EFG and EF2 in the eukaryote is basically EFG in the prokaryotic and 2, it is in the eukaryotic, right? Are the essential for the translocation process, right? During the elongation phase, the protein synthesis, therefore, that's why the A as well the B, 2, that option is correct over here. Okay. Question number 6 says, this medicine prevents translation from the starting. Options are ricin, B is tetracycline, and C is streptomycin, D is cyclohexylamine. So our correct option over here is nothing but option number C, streptomycin. What basically is streptomycin done? Streptomycin, which is basically binds to the ribosomal subunit, right, in the bacteria cases, and it interferes with the bind of the ribosome too. So it is preventing the formation of initiation completion. Okay, so correct answer over here is nothing but option number C, streptomycin. Okay, remember this question is really important for your exam students. So moving toward question number 6, 7, this is not a necessary component in the translation. Option A is amino acid, B is ligases, C is mRNA, and D is anticodon. So as you all know that answer is very simple, question is very simple. So our correct answer over here is nothing but option number B, ligases. Okay. In the translation, what is amino acid as well mRNA? mRNA is very important factor in the translation, anticodon as well. But it is that in translation ligase is not very important. So option number B is correct answer over here. Moving toward question number 8. This specifies a specific amino acid and the tRNA molecule that corresponds to it. Option A is topoisomerase, B is RNA, then C is ribosomes, and D is tRNA synthetase. So correct option over here, my dear friend, is nothing but the option number D is tRNA synthetase. What it is basically, tRNA synthetase is also known as the amyl acetate tRNA, okay, synthetase. Amino acetyl tRNA synthetase is basically, it is an enzyme, right? Play a very critical role in the translation process during the translation, and that means during the translation the process is known as the protein synthesis. So correct answer over here is tRNA synthetase. Moving toward question number 9, protein synthesis is the process of creating new proteins. Option A is replicating DNA needed for the protein synthesis, the synthesis of amino acid from mRNA, mRNA synthesis from a DNA template. Option number D is direct synthesis of a amino acid from the DNA template. So correct answer over here is nothing but option number B, the synthesis of an amino acid from the mRNA. As you all know that it is very important, the synthesis of the protein acid from the mRNA, right? As the process involving the assembling amino acid into the polypeptide, which is the based on the sequence coded in the mRNA, right? So here is the synthesis of the amino acids from the mRNA is correct option over here is. So question number 10 is amino acids having the hydroxyl group included the following. Option A is serine and alanine, B is alanine and the valine, C is serine and threonine, D is valine and isoleucine. So as you all know that the question in the question number 10, the correct option over here is nothing but the option number C, serine and the threonine. Serine and the threonine, which is the basically contain the hydroxyl group in its side chain. Okay. So option number C is correct answer over here. Rest of the all which is not at the side chain hydroxyl group is involved. Okay. Question number 11. The 21st amino acid is option number A, hydroxylysine. Option number B, hydroxyproline. Option number C, selenocytine. And D is nothing but the citrulline. So correct option over here is nothing but option number C, selenocysteine. So selenocysteine is also known as the 21st amino acid, as you all know that. Okay, and it is basically unique among the all standard amino acid, right? Basically, as it is a unique in the all standard amino acid, basically the selenium content in them, sulfur content in them, cysteine content in them is found, right, in the selenocysteine. So correct option is option number C. Question number 12 is, which of the following amino acid is a positively charged? Here question is positively charged. Okay, so option A is lysine arginine, B is lysine asparagine, C is glutamine and arginine, D is lysine and the glutamine. So correct option over here is option number A, lysine and arginine. So basically the the positively charged means what? It is an amino acid having the side chain and it contains the additional amino group, means the positively charged, which can accept the proton and thus carry the positively charged as the psychologically pH. Having the pH is around the 7.4, because most of the amino acids it is active around the neutral range. Okay, so positively charged it is having the lysine and arginine. Question number 13 is, which of the following amino acid contain the sulfur? Option A is cysteine, threonine, B is methionine and threonine, C is cysteine and threonine, D is cysteine and serine. So correct option over here is cysteine and methionine. As you all know that the cysteine and the methionine, that is the amino acid which contain the sulfur amino acid, sulfur sulfur group in that side chain. Okay, question number 14 is, in the polypeptide chain, which of this is the initial amino acid? Option A is serine, B is valine, C is alanine, and D is methionine. So correct option over here is, as you all know, that is nothing but the option number D, methionine. Okay, see in the protein synthesis translation process, right? The initial amino acid incorporated into the growing polypeptide chain is methionine, right? So this is due to why? This is due to the role of start codon. Role of start codon, it is nothing but the AUG, which coded for methionine in the prokaryotic as well in the eukaryotics. So correct option over here is methionine. Question number 15 is, in the instant of an amino acid that produce acetoacetyl coenzyme A during the catabolism of its carbon skeleton, which of the following is true? Okay, question is regarding with the true factor. Option A is it is a glycogenic in nature. Option number B is it's ketogenic in nature, and option number C is it's a essential amino acid. D is can be either glycogenic or the ketogenic in the nature. As you all know that, okay, acetyl coenzyme during the catabolism carbon skeletal, which is in the option number B, it is the ketogenic in the nature. The amino acid basically that produce the acetyl coenzyme A, okay, that is directly involved in the synthesis of the ketone bodies to make the ketogenic. Okay, to make the ketogenic. So option number B is correct answer over here. This question is really important for your entrance exam student. So remember it out, revise it out. Okay. So question number 16. In the nature, which of the following amino acid is both glucogenic as well the ketogenic? Option A, leucine, lysine, isoleucine, and histidine. So correct option over here is nothing but option number C, isoleucine. Isoleucine is a both, right? It is a glucosenic as well it is a ketogenic. Because why? Because it is broken down into the blood. Glucose and the ketone. Glucose having the precursor, that is nothing but the succinyl coenzyme A, and in the ketone bodies the precursor is nothing but the acetyl coenzyme A and the acetoacetate, right? Yeah. Moving toward question number 17. Question number 17. What is an example of the amino acid? Option A is alanine, B is glycine, C is proline, and D is serine. Okay, so correct answer is nothing but the option C, proline. Basically, proline is only amino acid among the standard 20 that is classified into the amino acid due to the distinctive in the cyclic structure involving in the nitrogen atom. Okay, so option number C, correct answer. So question number 18 is the distinction between amino acid and the amino acid is 10. Option A, bonding of the carbonyl group with the amide group, bonding of the nitrogen in that amide group, binding of nitrogen in the carbonyl group, presence of the covalent bond between the amide and the carbonyl group. So option number B, correct answer here is bonding of the nitrogen in the amide group. Okay, the key difference is nothing but the bonding of the nitrogen group in the amide group. In the amino acid, right, the nitrogen is bonded to two carbon atom, forming the ring structure, whereas in the typical amino acid, the nitrogen is bonded in the alpha carbon and the two hydrogen atom. So correct option over here is bonding of a nitrogen in the amide group. In the amide group. Question number 19. Which of the following amino acid is an important one? Okay. Option A is cysteine, asparagine, glutamine, phenylalanine. So our correct option over here is option number D, phenylalanine. As you all know that the phenylalanine, it is under the category of the cannot be synthesized. Okay, means we can fulfill that requirement through the diet. So correct option over here is the very important one amino acid is nothing but the phenylalanine, which is the very essential amino acid category. Question number 20 is which of this amino acid is a non-essential amino acid? Okay, non-essential amino acid is those which is synthesis in our body. Okay, which can't be we take it through the diet. Okay, options are lysine, threonine, serine, and the histidine. So option number C is correct answer over here is serine, which is synthesis in our body as well. It is under in the category of the non-essential, right? So moving toward question number 21, which of the following statement is the correct one? Okay, is the correct one. Option A is tryptophan and the tyrosine are the significantly more polar than the phenylalanine. Leucine is a commonly used as the ingredient in the buffer of the C SDS page. Aspartate is an essential amino acid. Lysine is a non-essential amino acid. The correct answer is nothing but option number A. Tryptophan and tyrosine are significantly more polar than the phenylalanine. Because why? The relative polarity of the relative polarity of these amino acid, okay, tryptophan and tyrosine, presence in the polar functional group, okay, in the side chain of the tryptophan as well in the tyrosine. That's why option A is correct answer. Question number 22. During the catabolism, which of the following amino acids produce acetyl coenzyme A? A is ketogenic, glucogenic, essential, none of the above. Correct answer is option number A. Okay, option number A, ketogenic. Ketogenic amino acid produce the acetyl coenzyme A during the catabolism, which can be used energy for the production of the citric acid cycle as well for the synthesis of the ketone bodies. Okay, ketone bodies. Moving toward question number 23, aromatic side aromatic side chain are found in the amino acid. A is tryptophan, asparagine, tyrosine, tryptophan, threonine, tyrosine, phenylalanine, tryptophan, serine, phenylalanine, tryptophan, and the tyrosine. As you all know that it is a very simple question. Option number D, phenylalanine, tryptophan, and the tyrosine is correct answer over here is which contain the aromatic side in their structure. Okay. Question number 24 is the non-polar aliphatic R group is found in the amino acid. Option A is glycine, alanine, leucine. B is serine, threonine, cysteine. D is lysine, arginine, histidine. D is phenylalanine, tyrosine, and the tryptophan. So correct option over here is nothing but the option number A, glycine, alanine, and the leucine. It is having the non-polar in the nature as well the aliphatic side chain that contribute basically the hydrophobic characteristics of the protein, right? Hydrophobic characteristics. Option number A is correct answer. Question number 25. Amino acids are typically produced from which of the following components? Fatty acid, alpha. Question number 25. Amino acids are typically produced from which of the following components? Fatty acid, alpha, ketoglutaric acid, mineral salts, and volatile acids. Our correct option over here is, okay, that is option number B, alpha ketoglutaric acid. Okay, it is a very crucial intermediate in the citric acid cycle. Okay, as well serve as the key precursor for the biosynthesis of the several amino acids. Okay, and which is really activated in the transamination reaction. Okay, so our correct option over here is the alpha ketoglutaric acid. Okay. So we have completed the question. This is our application, FoodTech Online. This will help you prepare for the exams such as FSSAI, GATE, State FSOs. It covers all the major subjects like the food processing, food chemistry, food engineering, food microbiology. It will prepare you thoroughly for your competitive exam as well, and it has a good number of the sets question for your practice. So please go through this app, and link of this is given in the description box. So join our app. Thank you so much for the attention, and please subscribe to our YouTube channel and also join us on the social media, link of which given in the description box. All the best student for your entrance exam.